Now we're going to talk about the development of the central nervous system, and we'll start with the hindbrain, the most simple part of the brain, and work our way up to the cerebral hemispheres. So by way of reminder, brain development really begins with the formation of the neural plate way back at roughly week 4 or so. And remember how when the neural plate was forming, it sort of got these little dents to it, creating these roughly sort of 3 sections of the neural plate. And those 3 sections actually correspond to the 3 major regions of the developing brain: the prosencephalon, or forebrain, the mesencephalon, or midbrain, and the rhombencephalon, or hindbrain. Now the neural tube will actually expand within each brain division to form sort of 3 primary brain vesicles, which will ultimately go on to develop further. Now, by the time we get to week 5, we've seen a little bit more division within the brain. The mesencephalon has enlarged. The rhombencephalon and the prosencephalon have divided into 2 separate structures. So the rhombencephalons become the metencephalon and the myelencephalon, and the prosencephalon has become the diencephalon and the telencephalon. The brain tube has also started to fold in 3 different locations called flexures, and those are shown here. That folding is ultimately going to create the final shape of the brain. Now, the ventricles of the brain are formed from the neural tube, which is sort of running through the center of this still somewhat tubular brain structure, and we'll talk about how they ultimately form and develop going forward. Now, within these developing brain structures, we have development of neurons. The cytodifferentiation, or the cellular development or differentiation of the neurons, comes from the neural tube, but primarily in the region of the rhombencephalon. So those are the ones with the blue background here. First, you get neurons developing, followed by the glial cells, the support cells, so the astrocytes, oligodendrites, epipendymal cells. All of those are produced after the neurons. Now, we'll start with the spinal cord and then sort of work our way up here. The spinal cord is much simpler than the brain, but the basic structure is relatively the same. So there are 4 plates that are derived from the mantle layer of the spinal cord. And those are the neurons that are derived from the neural tube, and then they migrated into the periphery of the spinal cord to become the gray matter. So that mantle layer subdivides into 4 plates that run up and down the length of the spinal cord. There are 2 dorsal plates and 2 ventral plates. The dorsal plates will become association neurons. You'll see pictures of them later, but basically they communicate between sensory and motor neurons. So they're not really sensory or motor neurons themselves, but they link those 2 neuron groups together. The ventral plates become somatic motor neurons, and again, those are the neurons that are going to innervate the striated voluntary muscles. Motor neurons tend to form first, followed by sensory neurons, and we'll talk a little bit more about that later. Also, we see the intermediolateral cell columns, and those are shown in yellow here. They contain the visceral or the autonomic motor neurons. So these are the ones that you don't control on your own. In the thoracic and lumbar regions of the spinal cord, that'll be the sympathetic autonomic motor neurons, and in the sacral regions, it'll be the parasympathetic autonomic motor neurons. So from the spinal cord up to the brain. Again, when we think about the brain, we think about higher centers, the cerebellum and the forebrain, although we'll talk about the cerebellum when we talk about the brain stem because it derives from the same structures. And then the brain stem, which includes the myelencephalon, the metencephalon, the pons, and the mesencephalon, which are all shown here. So again, brain stem, similar in development to the spinal cord, but more complicated. Instead of 4 columns, there are 7. And those 7 columns go on to do a somewhat more diverse array of functions than in the spinal cord. So here we have the cranial nerves. We've got efferent nerves, where the cell bodies are based within the spinal cord, and those are providing outgoing motor functions. So essentially, if you're based in the spinal cord, you're part of the process that sends signals back out into the body. Whereas your afferent neurons are providing, again, motor functions in this particular example, but they're the ones that are sort of entering the spinal cord. Now, within the brainstem, we start to talk about the cranial nerves. These are derived from these columns that we looked at in the last slide, but not all of these columns are present in every layer of brainstem. So we've got sort of the distinction of the efferent neurons on one side, the outgoing neurons, and the afferent neurons or the incoming neurons on the other side. And what you'll see is that there are regions of the spinal cord up into the brain that don't contain all of the columns. And in fact, in the mesencephalon, there are really only 2 columns present. Now, when we're forming the brain stem, again, this forms from the rhombencephalon. What we're showing here is that initially, the sort of early development of the rhombencephalon sees the roof plate, so sort of that top portion of the rhombencephalon, widening fairly significantly and forming what's called the rhombic lip at its dorsal edge, okay, on the sort of the back end. The neural canal of the rhombencephalon, so essentially the neural tube that's running through the middle of it, creating this empty space in the center, that's going to become the 4th ventricle. And then there are going to be openings that form that allow the 4th ventricle to connect with other ventricles. Within that ventricle, we're going to see the development of choroid plexus. Those are small sort of finger-like projections that sort of extend from the roof plate and produce the cerebrospinal fluid. And we'll actually see them in all of the ventricles as they develop. What structures derive from the rhombencephalon? Okay, again, we get the myelencephalon, which will become the medulla oblongata, and then the metencephalon, which will become the pons and the cerebellum. The cerebellum is derived from actually both the metencephalon and the rhombic lips that were formed we talked about on the last slide. And in the 3rd month, what we'll see is that there are 2 cerebellar hemispheres that have formed again from the combination of the metencephalon and the rhombic lips, and they'll be visible, and they're connected by cerebellovermis. Now, those hemispheres are going to go through many, many subdivisions. First, they'll develop lobes, and then lobules, and then gyri, much like the cerebellar cortex of the brain, or pardon me, the cerebral cortex of the brain. That involution, that creation of all of these different little projections and wrinkles, essentially, creates this, just creates an extreme increase in surface area. And the more surface area you have, the more neurons you can have. So the cerebellar hemispheres go through many, many different subdivisions, essentially creating all this extra surface area, allowing for more neurons. Now, within the cerebellum, the ventricular layer, which is shown in sort of pinky red here, and then the external germinal layer, which is blue on the exterior, give rise to the neurons of the cerebellum. So the neurons of the cerebellum form in these 2 structures and then sort of migrate inwards, laying down the first wave sort of at the very edge, right sort of where they developed, and then each successive layer going deeper. Now, the mesencephalon. The mesencephalon, again, is the caudalmost, or sort of the end of the rhombencephalon that's closest to the spinal cord. It differentiates, again, into dorsal and ventral columns that produce 2 of the cranial nerve nuclei, so for the oculomotor nerve and the trigeminal nerve. Also through the mesencephalon, we see a lot of neuronal connections between the forebrain and the hindbrain and the spinal cord. So again, this is sort of midbrain region, everything that wants to go from the hindbrain and the spine up into the brain and then back down again passes through here. Additionally, in this region, the neural canal, so the neural tube, the empty space there, is going to narrow and become the cerebral aqueduct, which is the passageway between the 3rd and 4th ventricles. Now, the cerebral aqueduct is critical for the normal function of the brain, but it can become narrowed or even blocked. And if it does, we get backup of cerebrospinal fluid into the third and then the lateral ventricles. Again, because those ventricles are producing their own cerebrospinal fluid, but the only way to get it out is to go through the cerebral aqueduct. And if it can't get out, you get what's called hydrocephalus, where the fluid is accumulating in the ventricles and ultimately pressing on the surrounding cerebral cortex. And in the case of a developing fetus, it may be compressing it to the point where it's not developing normally. Now, what you'll see in this picture, which is kind of interesting, is that the cerebral aqueduct is supposed to be formed by roughly 15 embryonic weeks of pregnancy, which means hydrocephalus, if it's caused by a congenital anomaly of the cerebral aqueduct, so something that's happening during its development, can actually start reasonably early in gestation, reasonably early in a pregnancy. And it is something we can see on those second trimester scans at 18 to 20 weeks in some cases. Sometimes, of course, it develops later, and then we won't see it at that time. Now, by way of review, there's this handy table here which nicely summarizes sort of from the very beginning, early stages of neural tube development, all the way through development of the brain and the ventricles, which structure leads to what, and I thought it was handy, so I put it in here. So we just reviewed development of the rhombencephalon, going through the metencephalon and the myelencephalon to form the pons and cerebellum, the medulla oblongata, and the fourth ventricle, and then also that the mesencephalon develops into the midbrain and the cerebral